All right, welcome back, everyone. We're going to go ahead and get started. We have about 45 minutes, and we have a lot to cover. The first talk is on Enroll HD, and I'm really pleased to welcome Jamie Levy from CHDI. Jamie's been working with CHDI since 2005, where she currently co-leads the clinical platform group that runs the Enroll study. She also manages the European HD Network, a nonprofit clinical network funded by CHDI Foundation, where she's primarily responsible for the business and administration aspects of the network. She's also an HD family member. She sits on the HD Family Council at Terrence Cardinal Cook Healthcare Center and participates on a New York State HD task force, and is on an advisory committee at the Center of Research on Ethical, Legal, and Social Implications of Psychiatric, Neurologic, and Behavioral Genetics. That's another mouthful. No acronym for that, which is a joint project of Columbia University Medical Center. So with that, I want to turn over to Jamie Levy.
Thanks, George. So thank you very much. I really want to say that this month is the 5-year anniversary of Enroll. And I know many of you know about Enroll and many of you have participated. And we can't thank you enough. So you can see from this line, this is what global recruitment in Enroll has looked like over 5 years. The very bottom of the line, that is about, I think it's around 800 participants at the end of the first 12 months. This month, I think only from a few days ago, we're over 14,000 participants from HD families. So really, thank you, because you're making this study a great success. So what is Enroll? I can't be presumptuous to think that you all know what it is. I know you've been hearing about it for years, but some of you may never have heard about it until today. So it is a global research platform which aims to speed up progress in clinical trials, to understand more about the disease, the phenotype, the genotype, and also to improve care. It has 3 main goals: to improve the understanding of HD and find what factors influence the progression. I'm not sure why 2 went before 1, but the main thing is to enhance the design and expedite the conduct of clinical trials. And I'll tell you a little bit more about that in a moment. And also to foster good clinical care and improve the health of the participants. So who participates? Enroll HD is a family study. I hope those of you that are participating invite your family. It can be someone who is gene positive, carries the gene, someone who knows they have the gene, someone who doesn't know their genetic status, gene negative, spouses, partners, caregivers. So you can come to the study as a family and participate, because all of your information is quite valuable to help recruit for trials, to run studies about Huntington's disease in the lab, and also to help clinical practice. So what is involved in an Enroll HD visit? It's almost like a regular clinical visit. You go to your doctor, they'll take your medical history, they'll run a few assessments on cognition and behavior and motor, we'll take blood and ask a few other questions about how you're faring, how you're thinking, for caregivers, how you're feeling, how are things at home. And what does Enroll collect? What type of information are we gathering? It collects a common set of information for all participants, from all the sites around the world. We presently have about 140 study sites in many countries. I think we're up to about 15 countries or 20 countries. I've lost count. We expect by the time startup, which is still, believe it or not, in startup, we've been running for 5 years, but it takes time to get sites started up. And we expect to be completed with the startup phase by the end of this year, middle of next year. And at that point, we expect to have about 220 study sites conducting Enroll. It also collects blood samples for research purposes. This is not only the genotyping, which is research genotyping only. It also is, we ask for optional samples so that we can run other studies. And it's not only for CHDI, it's for any researcher that wants to access this information and these samples to conduct studies. You're probably aware that all the data in the samples are coded, and this is to protect the identity of anyone who participates. So data sharing is one of the most important features of Enroll HD. It's essential to develop therapeutics for Huntington's disease. So not only are there research studies conducted in labs looking at biomarkers or looking at anything that may stop the progression of the disease. But it's also used for helping design and develop protocols for clinical trials. Because the information that you contribute by coming to your own Enroll visits is remarkably valuable for helping the clinical trials get started. The coded clinical data and biosamples collected from the participants are made available to researchers through a very straightforward process. And you can go to the enrollhd.org website and look at the process. You just click, you can see all the studies that are presently being conducted. So Enroll helps also to recruit into clinical trials. This funnel shows you what it takes to get people recruited into clinical trials. We've assessed, just from past experience, Enroll has helped present lists of potentially eligible participants to the study sites, so that the study sites that may want to recruit their participants for the clinical trial, they know who to look for based on the criteria that's in the clinical trial protocol. So this is just to show you what it takes and why we need so much data and why we really are enthusiastic and encouraging participation into Enroll. In the last 3 clinical trials that Enroll has helped provide data that you've contributed, it took approximately 8,000 data sets or 8,000 participants to come up with 800 participants who were suitable for the trial. So it's almost like a 10-to-1 ratio. So it's really important that we gather all this data and are able to, you're counted in a way so that you can be contacted by your site if you choose to be contacted for a clinical trial. And how does Enroll improve clinical care? Well, we identify best practices across the Enroll study sites and work to ensure that the standard of care provided to HD families is consistent. We recently launched an HD clinical trial portal, which is for training for the sites, for the raters, for the PIs, but it provides a consistent way of training so that the results, it minimizes the variability of the results. And if everyone's trained in the same fashion, then the results will be of the best quality possible. So we were very pleased to launch that, and there will soon be other modules as part of the training portal to help your doctors, your sites, your raters, your PIs to participate and enroll. So we've been up and running for 5 years, 14,000 participants. We expect by the end of 2018 we'll have about 20,000 participants when all the sites are up and running. And now we have to take a look and say, wow, this has been phenomenal. It's happened sooner than we actually expected. And now we want to try to focus on, do we have the type of data we need? Are the participants really able to come to visits regularly? Are we honing in on the right group of participants, the right type of family members to participate in specific trials? So we've assessed that there are some things we'd like to change going forward. It's going to be the same Enroll. You come in for your annual visit. But what we'd really like to do is increase the recruitment of the at-risk people. That's people from HD families that do want to participate in research, but that do not want to know their genetic status. There is the genotyping, but it's only for research. So we encourage people. We'd like to get people that are at risk, that are pre-manifest, and of course at the early stages of the disease. Because as you heard or may be hearing later, that a lot of the clinical trials that are seeking recruitment into them, they're looking for people in that particular stage, whether it's early stage or a little bit earlier than being manifest. So it's our job to be able to collect data and samples from the HD families that really fit that criteria. So we're really hoping to encourage those people from HD families that would like to participate in research to come with your affected parent or family member and join the study. Another thing we're looking at is that as people progress through the disease, it's a long visit. I mean, well, long. It's 1 to 2 hours. But also, as you progress through the disease, not all people can handle all the cognitive assessments. I know, because I was with my sister when she participated, and it was very frustrating to see her try to do these cognitive tests. So what we're trying to do is transition all people, as they become more advanced, into a lighter, easier protocol. We haven't come up with a name yet. Enroll Plus, maybe, Enroll Light, but the idea is that we have an easier protocol for people as they go into a more advanced stage of the disease, allowing them to transition from the full enroll to a shorter, easier, simpler enroll. The third aspect that we'd really like to focus on is taking advantage of mobile health applications. We feel that if we can take advantage of what's happening around the world really with mobile health application, whether it's a wearable or a sensor or a smartphone or an iPad, that a lot of these assessments can be conducted online at home. And we also feel that we'll reach out to people that don't want to come to clinic all the time. Either they're younger and they're not interested in coming to clinic, or they're working and they can't accommodate coming to clinic on a more regular basis, or they're remote and we can't reach them. They can't get to clinic. So we're broadening our net, and we're hoping that with the launch of mobile health applications in the next couple of years, that we will be able to attract a broader cohort, broader amount of family members that'll participate in Enroll. So these are the three items that we wanted to make you aware of. You'll start hearing more and more about it as we develop the protocol. The actual change in the protocol is still probably a year or two away, but you'll be hearing more about how we're going to change the protocol, and here you're seeing through the three main ways we intend on doing so. So what can you do? Make Enroll your study. Be informed. The enrollhd.org website is there. I'd like you to go take a look when you can. You can see all of the ways your data is being used. We have data cuts approximately once a year, and any researcher that comes from an institution, we don't take Gmail or Yahoo addresses, but if you're from an institution and you want to research using the samples and the data from Enroll, you just have to apply. And you, as the community, can go online, you can see all the projects that are currently being undertaken using the data and samples you've contributed. There are presently over 100 studies being conducted with your data and samples. And for that, we really want to thank you. And you should give yourself a round of applause, because that's an enormous amount of research. So pass on the word, recruit and enroll, be a champion, and get involved. I'd like to thank the HD patients and families. We couldn't be here without you. The study sites, the PIs, the study coordinators who do phenomenal work organizing their sites, the raters, the neuropsychologists, everyone who you are working with at your study sites. The patient advocacy groups, the HDSA does a remarkable job. It really does take a village. It's our staff, the governance committees, CHDI who supports the study and employs all of us who work on the study. It really is a community effort, and it's with a platform like Enroll that we can expedite the conduct of clinical trials. We can facilitate the enrollment into clinical trials. It's speeding up the process. So thank you very much for your attention, and please get involved in Enroll.
I recognize we have about 30 minutes, so if you have questions, if we have some time at the end, we can ask questions of the three speakers. I'm going to move on to the next presentation, which is Paul Bono from Wave Life Sciences. Dr. Bono joins us from Wave in Cambridge, Massachusetts. He has over 15 years of scientific and strategic leadership, and prior to joining Wave as the president and CEO, he was VP of Worldwide Business Development, head of Asia Business Development Investments, as well as head of Global Neuroscience Business Development at GlaxoSmithKline. He's earned a medical degree from Hahnemann School of Medicine and an MBA from Drexel University. He was a general surgery resident and cardiothoracic surgery postdoctoral research fellow at Drexel University College of Medicine. Please welcome to the stage CEO of Wave Life Sciences, Paul Bono.
Thank you, George. And just to start, I'd like to thank the HDSA. This is actually a really important moment for us at Wave, because this is our first presentation at a clinical trial session. But it's important to know that we're still a preclinical company and about to make that transition shortly. So thank you, and thank you for letting us, and I'm going to try after Ed and Jeff's speech to make a slight amendment here, but thank you for letting us share the progress on developing our allele-specific mutant Huntington protein lowering therapies with you. So we're going to get better, rather than calling it silencing, to call it lowering. But it's important because, as Elena shared this morning, we're a scientific-driven company. And so we started the approach early, recognizing that we're standing on the shoulders of giants. People have been involved in the field for a very long time, and we were able to benefit from a lot of the scientific advancement to drive our discoveries within WAVE. So to start, and you saw some of these slides earlier, we recognize that there's this relationship now based on the Huntington gene. And if you think about the gene, I always think about a gene as an instruction booklet. It's a book with a lot of information in it. But it's the RNA, so this messenger RNA that you saw earlier, that I view as kind of the word. And within the word, that can mean things. So in the case of the body and biology, what it means is actually this protein. And two things can happen. Either changes can happen that take letters out of the word, in which case you can't understand it and something's missing, or you can add additional letters to the word, and the word becomes more complicated and translates to something that it shouldn't be. So in the case of the Huntington's disease, what you see is the addition of these additional letters, the translation of that to this protein that becomes elongated, and therefore it causes damage. So the recognition of antisense therapies, and you heard the word about antisense therapies before, is the recognition that you could make a medicine that potentially recognizes that messenger RNA, the word, and by binding to that can actually remove the word, and therefore remove the protein. What's unique about the approach that we're taking at WAVE is that we can target that ASO, that antisense molecule, specifically to the mutant protein, leaving the healthy protein intact. And as we learned earlier, there's good reason to believe that the protein is there for a reason, and the more you can leave intact, the better, and therefore focus on silencing or therefore lowering the mutant protein. So how do we do this? We do this through special chemistry. So this field's evolved a lot in the ability to make these medicines, as we saw recently with the Ionis study, the translation of that into therapies. What we've evolved is the chemistry, how we get those medicines into cells. And there's something that's important, that a lot of the advancement in this field that went to the chemistry that made this happen put something called a chiral center, this left hand, right hand. So if everybody puts their left hand and right hand together, you see that it's like a mirror image of one another. But if you then try to put your left hand on top of your right hand, you realize that it doesn't overlay perfectly. And that's a lot like a baseball glove. Drug development, making a medicine is a lot like sticking a hand in a baseball glove, right? The glove represents this protein that you need to bind to, and if you have a right-handed glove and you stick your right hand into the mitt, that's great. It fits really nicely. But if you take your left hand and you stick your left hand into, and I don't know if anybody's ever tried this, into a mitt that doesn't fit, you see that it doesn't get in there. And the recognition in all of these different therapies are that the current drugs right now are a mixture, in some cases, of over 500,000 different drugs that you're relying on to fit into that perfect pocket. So what we've been able to do at WAVE is actually develop the chemistry that locks that left hand and right hand into place, thereby helping that hand, if you will, that medicine, fit more preferentially into the glove. And what does that do? What that does is it enables us now to have single drugs that we'll use in our therapeutic study. It means we can characterize the drug in terms of how well it's engaging with its target, in this case, lowering the protein. We can look at safety signals and see its avoidance. So in some cases, you don't want it to fit in the glove, and design how not to have it fit in the glove to cause unwanted effects. And so this ability to fine-tune what the aspect of the medicine that you're giving is gives us this unique insight into how we translate our medicines into therapies. So this is great if you know what your target is. So what's unique for WAVE is we had to overcome another targeting challenge, which is what is the difference between the allele, so the gene one has from one parent versus the other, one parent having Huntington's, the other not. So inheriting the CAG repeat versus the other allele that has fewer CAGs. And what we realized was there's some interesting insight that there were actually, I like to think of it as some of us have used pins on our phones for genetic GPS. And this idea that if you look at this sequence of various, these alleles, these genes, that one can find this kind of homing, this GPS signal on one that's not on the other. The ability to do that control, that left hand, right hand, I said, allows us to cut in one particular place when it binds. So marrying these two concepts together, we could develop a medicine that targets that genetic GPS, but doesn't target the other that doesn't have it. thereby having a customized therapy that actually just removes or silences or reduces the mutant protein. We've developed, because we've been able to, 2 programs, and we'll talk a little bit about those 2 programs, but 2 programs. These signals, what are called SNPs, you'll hear that word a lot. These SNPs are those, what we're calling these genetic GPSs. We're developing 2 therapies that each target a different SNP and are currently running an observation study to look at the prevalence of this SNP1 and SNP2, trying to, again, ascertain how we can treat more patients. Right now, these 2 programs are present in about 66 to 70% of HD patients. So as we develop this, it was important that we see this separation. So in these studies that we've done using patient cells, we've been able to look at what's the dotted line, which is where you can't control that left hand or right hand, or the ability to develop a therapy where we do control that left hand, right hand, the solid lines. And what you can see on your side to the left is that we see a separation, this ability to be able to do what we call dose response, being able to give more drug and see a separation between how we bring down and lower that mutant protein in blue and how we're able to resolve and leave that healthy protein intact. Now, that's one side of the coin. As we said, sometimes you want your hand to fit in the glove and actually engage the mechanism to silence. The other side's equally important. There's certain gloves you don't want the product to fit into, your hand to fit into, and that's immunogenicity or activating the immune system. So it was really important as we fine-tune our product to be able to not have it actually activate the immune system. So in the case of the slide on your right, the green line's water, the blue line is the compound that we designed, and the stereorandom equivalent is that gray line. So again, being able to fine-tune the program was a key aspect of how we developed this potential medicine. This slide is meant to highlight, we showed that line earlier, that there's this kind of, here's the word, and then ultimately what it means, the protein. And the ability to say that how we can discriminate, being able to knock down, so lowering that blue line, keeping that pink line as high as possible, and more importantly, being able to do that at a variety of doses, because as we translate this now from a preclinical program to a clinical program, it's going to be important for us to see that we can give more drug and not lose that discrimination between the healthy protein and the mutated protein. So we're developing that. One can imagine at this point in time, now we've run these studies in cells. We've translated a show in vivo studies that we can deliver this to various parts of the brain, so we see it get to the same areas or similar areas as our peers, so it goes to the cortex, it goes to striatum, cerebellum, the places where it needs to go. And we've run our preclinical safety experiments. And this is important because it leads us to the next piece, which is our translation to the clinic. So we will be embarking the middle of this year, which translates typically to summer, to running 2 global studies, one for each program, our SNP1 program and our SNP2 program. There'll be 2 placebo global controlled studies. It is important to us, as you heard earlier from both Ed and Jeff, that we want to run well-designed, well-powered studies. Experimentation is important, and it's important for us that at the end of this study that the data can tell us something. And oftentimes a placebo is the best way to see the results of that data on the other side. We too will have the concept of an open label extension built in for patients who are participating in the study, because it's an important component and we realize that everybody's contributing something to everybody's learnings around this program. The primary objective, because this is an initial study, will be safety. That's very important to us, that it's a safety study. However, as you heard, the advancement now in terms of biomarkers, both the ability to lose, and I can never do it as well as Ed around the laser capture mutant protein assay, so I won't even try, but the ability to look at the measurements of mutant protein in the CSF, the ability to look at some of the other emerging clinical biomarkers will be embedded in this study. We will have clinical measurements, and we'll also be looking at MRI imaging. A lot of the larger studies, TRACK-HD and others, are contributing significantly in ways that as we run this study, being able to have benchmarks, so real ongoing natural history studies are very important as one's doing drug development to have something to compare to. So we benefit from a lot of the work that's been done before on these observational studies. Patients will be identified as being SNP1 or SNP2, so a lot of you we've discussed around, I think a couple people in the audience, around how CAG testing gets done for Huntington's. We'll be adding another layer to that testing, which will be identifying SNP status, SNP1, SNP2. It'll be approximately 50 patients per trial, and when I say per trial, I mean 50 patients in the SNP1 study, 50 patients in the SNP2 study. Key inclusion criteria up there, but we'll definitely be moving into the early stage of the disease as part of this study. So we're very excited, and as Ed gave a preview to, we'll be coming up with the name. This study will be called the Precision HD Study, and we are very excited. More excited and I think gratitude and a lot of thanks as we're making this progress goes to a number of people here. It goes to patients, it goes to their families and caregivers, it goes to the clinicians and researchers who've been collaborators as we've been advancing these programs within WAVE. Your insights into the programs, your insights into our potential trial designs are critical and inspire us as we're on this journey. So thank you very much.
All right, so yet another clinical study coming. So I'm going to now turn it over to Dr. Neil Simon. Neil Simon is the co-founder and CEO of Azavan Pharmaceuticals, a clinical stage drug development company which has a current drug in the clinic called for Huntington's disease called the STAIR study. Oh, sorry, it's called it's a STAIR study. Dr. Simon brings 20 years of management experience and 30 years of research experience in neurobiology and drug development to Azavan. Please welcome Neil Simon.
Oh, good afternoon, everybody. I want to thank George and HDSA for inviting us to tell you our progress in the STAIR trial. And I need to begin with just some transparency. Azavan is a small private company, but I have an interest in the company. I'm a neuroscientist by training. I'm a pragmatist, but you should know that Mike Brownstein and I do hold equity in the company, and Eve Damiano is our senior vice president. She's an employee and receives a salary. So having done away with the formalities, I'm going to talk to you today about irritability and severe neuropsychiatric symptoms in Huntington's disease. But let me begin with a short story. So I don't have Huntington's in my family, but like many people in society, one of my parents had Alzheimer's disease. So my mother went from a formidable, bright, gracious, and unfailingly polite woman to a nasty, irritable, unmanageable person. I don't even think she knew what was going on, but it was hard for her, hard for us as members of her family, my father and my brother, and hard for her friends. You end up isolated, you become unmanageable, and frequently you end up institutionalized if you don't die before the problems become too severe. So irritability in psychiatry is it's an umbrella term. It includes anger, aggression, acting inappropriately, incorporates anxiety, agitation, and the like. So it's an umbrella term. And I can show you a technical definition. This was provided to us by Karen Anderson, who many of you know. You don't really need to read this. I think all of us know what irritability and anger means, if it's in our family or it's in our friends. And it can be episodic, it can be constant, it can be verbal, it can be physical, it can create a host of problems. And we hear repeatedly that this is enormously important to develop treatments for these symptoms. And irritability is actually the very first symptom to appear in Huntington's disease that we can recognize. So, what was the rationale for doing the STAIR trial? Because everybody has to have an acronym. We know that neuropsychiatric symptoms and irritability in HD really haven't received the attention they need. Most of the drugs we have are for movement disorders. It's seen, depending on who you ask, 40 to 70% of patients have it. It's the leading cause of institutionalization, makes people unmanageable, and frankly, I don't have to tell you in this audience, our approach to this technically is polypharmacy. We give people a whole range of drugs. Frankly, it's the kitchen sink approach. Nothing has worked. What we do is give people a bunch of things that create bad side effects. We make a bad problem frequently worse and increase their risk. So our compound, which targets the vasopressin 1A receptor in the brain, and I don't have time to go through all the science today, really is a new way to think about treating irritability. Vasopressin is an old neurotransmitter involved in social and emotional behaviors, and we know that if vasopressin function is increased in the brain, in humans, you end up with higher irritability, higher aggression, and higher anger. So it's a key signal in the brain for this. We know we're addressing the dominant receptor subtype. It's found throughout the brain and everywhere we've looked. And the drug, which is called SRX246, is the very first drug in this class. So it's called a new chemical entity, and it was the first of its kind developed. So to put 10 years into about 30 seconds, some of the key things we found is the receptor is in the brain, and it's in the brains of HD patients when we look at their brains post-mortem. So even with the neurodegeneration, it doesn't go away. We've demonstrated proof of mechanism for the drug in an fMRI study that I don't have time to go through. We've seen excellent safety and tolerability in all of our studies. And recently, which is very encouraging and exciting for us, we treated patients with something called intermittent explosive disorder. So these are individuals who periodically get set off uncontrollable rage and anger, can't keep friends, can't keep a job, can't stay in a relationship, frequently are in trouble with the law. And we saw clinical benefit to them. So they're not HD patients, but they have some of the similar symptoms we see in irritability. So we're quite encouraged by those results. So this is what the STAIR study looks like. We started in May of 2016 with our first patient. This is done in collaboration with the Neuronext network at the National Institute of Neurological Diseases and Stroke. We expect to enroll our last patient somewhere between October and December of this year. There are 22 sites. There will be 108 subjects in this trial. This is a very large trial for a Huntington's disease population. This is the first well-powered, rigorously designed study for neuropsychiatric symptoms in Huntington's disease that's ever been conducted. Everything else you've seen are case studies, one-off studies. I gave this person this and they got better. That really isn't the way good science is done, nor is it the way treatments are developed. So what I'd like to call your attention to, this is a dose escalation paradigm. Neither we nor the investigators nor the patients know what they're getting. But 2 people will get the drug for every 1 that gets placebo that comes into the study. And we start at 80 milligrams twice a day. That's based on the pharmacokinetics. Then after 2 weeks, the safety's been established. We escalate to 120. Some patients will stay on that dose for 10 weeks. After 4 weeks at 120, some will escalate to 160 milligrams a day. I can tell you right now, we've had no safety issues or tolerability issues thus far. We're blind. I can't tell you what's going on with the symptoms. So I should say something to you about inclusion criteria because we get lots of people that ask us about the trial. This is a summary list, but let me call your attention to 3 of the criteria. So first, we expect people to have symptoms of irritability and or aggression to enroll in the study. That we use a scale you're all familiar with, the UHDRS. People have to have a responsible study partner. Why? For those of you, and I'm sure it's many of you in this room, who deal with irritable and angry patients, you can ask them sometimes, what kind of day did you just have? And they say, oh, I had a really good day. And there's a very scientific response to that from caregivers and family members. Their eyes roll up into their head and they shake their head no, right? So we need a study partner to help us understand what the patient's behavior really is. And then the other thing that's important is we know that Huntington's patients are on many different medications. We are not taking them off those medications. We're adding ours because ethically you can't change regimens and also because our drug appears to be very safe, and that's what we've seen so far. So this is a consolidated reporting system for a clinical trials chart that we have to produce periodically. And what I'd call your attention to is right now 62 patients are in. We have 5 in the queue. I have to tell you, working with the Huntington's community is a privilege. People are very motivated. We've had 1 person leave the study after the 6th week. And the only reason she left was she had a 4-hour drive to the clinical site and just couldn't do it. So far we've seen the drug is very safe, very well tolerated. 33 people have finished, 8 more are in follow-up, and we need to recruit and randomize another 31 people to get done. So we're hoping to get to there, as I said, in the fourth quarter. So these are the clinical sites. There are 22 of them. I put this list up. This information is on clinicaltrials.gov. It's also on the HD Trial Finder. George was good enough to help us with that. If you know someone who might be interested in the study and lives near any of these centers, please have them contact the sites. Many of these are HDSA centers of excellence, also HSG centers of excellence. There's strong overlap. So what are we measuring here? Well, SRX-246 is what we call a new chemical entity. Our primary concern at this stage is safety and tolerability. Can you take it, and is it safe? The answer is yes, based on everything we've seen so far, although that's always an ongoing process. And you see there it says exploratory measures for irritability. That's because we don't have any scales that have been established for use in Huntington's patients. So we are testing a number of different scales with a sufficient number of subjects so we can identify the scales that are best used to measure changes in anger and irritability in a Huntington's population, and then use that information to go to the FDA for phase 3 planning. So at this point, we're encouraged by the safety and tolerability data. We're blind. I can't tell you about the behavioral measures. I need to acknowledge far more people than I could put on this slide, but we've had people help us from the federal government, particularly NINDS, which is paying for this trial. But what I do really want to recognize, and I'll say the same thing everybody else up here has said, HDSA, HSG, and the Huntington's community. It is remarkable working with you. Your commitment to developing treatments to help with the disorder is just astonishing, and it really motivates us all. So what I hope now is that in a year, I'll be here and be able to tell you about the results, and I'll be here with good news about the development of a new and safe treatment for neuropsychiatric symptoms in Huntington's. Thanks for your time.
All right, almost there, everyone. I think we're just going to flip through these. These were some backup slides. So we've heard from Jamie, we've heard from Paul, we've just heard from Neil. Before that, Ed and Jeff. And actually, after this session, if you want to hear about more exciting research, the folks from Shire and Sangamo are going to be talking in Schaumburg A&B about their zinc fingers, as well as PTC Therapeutics about a small molecule to lower Huntington. There are lots of great approaches coming, and we want to be able to share how to get involved if you're interested. So this is just, I don't have to belabor this point, but it just bears repeating. Without any volunteers, there will be no trials. With no trials, there will be no drugs for Huntington's disease. It seems obvious, but I just have to repeat it. You can see the numbers here. 80% of all clinical trials, not just Huntington's trials, but all clinical trials fail to finish on time. We don't have time for that. So we need to get involved. How can you get involved? Well, you can, and I hate belittling clinicaltrials.gov, but it is an option. There is a lot of information there. A lot of it is very technical. If you like to drink from a fire hose or look for a needle in a haystack as a pastime, this is a great place to go. If you type in Huntington's disease in clinicaltrials.gov, you'll see that there are over 1,000 opportunities for you. There are not 1,000 clinical studies that are recruiting for Huntington's disease. That was a problem. So we saw that as a problem. So HDSA created a new resource, a new platform, an online source, as well as a call center. It's a little bit more civilized. I put this little picture in for Ed, but he's probably not here. It's called HD Trial Finder. You can get there at the hdtrialfinder.org website or on hdsa.org. And it provides you, it's a matching service. You put in a little bit of information, and you'll be able to, with your zip code, the status, answer a few questions, create a profile. You'll be able to match to clinical trials in your area. We recognize also that some people might not be really comfortable with putting some private information on the internet, may not have access to internet. We get those calls all the time at HDSA. So we added a call center. There's someone that's trained by HDSA. There's 3 of them. They're in North Carolina, trained by me and other people that are there to take your call Monday through Friday, 9 to 5, 866-890-6612. And they'll help you with the process. And in addition, if you're having problems contacting a clinical site, call them. And they call me right back, and I'll help you connect with the local site. It's super easy, as I mentioned. You go in here. This is kind of what you will see when you first try to set up an account. It's you create an account, you create a profile, basically a zip code, and who it's for. It could be for you. It could be a doctor creating it for a patient. It could be for your loved one. You answer a very short questionnaire, 11 questions. You can opt out of any of them. The more questions you ask or answer, the better the matching. And then finally, you just view your personalized results. And it might be very hard to see, but hopefully you'll pick out your phone, get on your computer when you get home, log in and take a look. This is for me as someone in New York looking for different clinical studies of my age that I match to. And what's really important here is you're going to get you're not going to get a 1-800 number for some place in India or China and have some non-personal information. You're going to get a name, a number, an email of the local site coordinator in your area that you need to reach out to. And we work very closely with Wave and Enroll and Azavan and all the other companies that are in this space to make sure that this information is as up to date as possible. What do you need to know? What's in there? Please take a look. It's super easy. You can't get messed up in there. It's in this database are only studies that are currently recruiting. So you heard some from Pride and the Amaryllis study from Pfizer that recently ended. You won't see them in this database because they're not options anymore. Not that they weren't important. It's just not an option. They're not in there. They're in clinicaltrials.gov, but they're not on our website. You're going to see observational and interventional studies, surveys, places to donate samples from blood, plasma, CSF, like HD Clarity. There's also really important experimental medicine studies that are being done, biomarker studies that you heard about from Ed with his neurofilament light protein recent paper. These are really critical studies that need to get done. They're not listed in other websites like clinicaltrials.gov, but they are super important that we get volunteers to participate in them. So please take a look there.